Do you see the PowerPoint slide downtown? Yes? Okay, we're disjointed here somehow. Function, this should pick up the... No, I've got that on computers. The internal. Okay, technology, do what you're supposed to do. I know what it is. Okay. This room is set up for computer science. With a click on this second monitor at the front here, the computer science teacher can actually, and this is how I'd log on to see what activity there is at any local computer. Today's fall convocation was about civility in the classroom, which kind of went with that lecture that I was went with the introductory lecture that we had on Monday about the requirements for civility in the classroom according to the syllabus. So it was refreshing to see that the entire university is backing that up. So keep in mind that we are in an academic setting. We do have professional commitments to ourselves and to others. And that respect in the classroom like this, where people have access to World Wide Web, is especially critical because the web is loaded with things that are distracting and offensive to many individuals. So we ask that you sustain I'm just going to set the camera here. Lighting isn't so good. Okay. Well, you don't need to see me that so much. All my tie and everything anyway. So much for the technical wizardry. We had discussed in the previous lecture the growth and maturation, and that it included senescence and aging, and I talked about a variety of things from the previous slide of the determination of where things are supposed to appear, and that I'm glad that I don't grow toenails on my antitragus of my ear, although it'd be easier to clip them if they were there. But I did, just before leaving this morning, check and had a few little hairs to pluck, and that reminds us of the senescent part of that. And actually, under the category of death, there was a moment of silence at the convocation today for two deaths, the son of a faculty member on campus and the chair of the Department of Psychology. So we are constantly reminded of all of these. And Jennifer corrected me at the end of the last class. She's a little more than just 6 weeks pregnant, which we were going to just wait and see if anybody could correct that on their own. But you may have noticed that if she was 6 weeks and showing like she's showing, there's a lot of rapid growth going on there in that embryo. So this brings us to the discussion of the human life cycle. And again, our efforts are to come up with clearly stated definitions so that we utilize specific terminology or general terminology and recognize that we're making an all-encompassing reference at that time. So the example was conceptus versus zygote versus embryo versus fetus, and what are the differences there. So embryonic is the zygote, the germ layers system. This is the earliest portion. It is the embryonic stage of the human life cycle, even though the zygote is not an embryo. It is the stage before the embryo. The fetal stage, we said it is the 8th week is when this begins, and so there's going to be further growth and maturation. So the embryonic stage is that stage, as we said, of cytogenesis, histogenesis, and organogenesis, and the full development, establishment of the organism. And we left organ systems out of that level before. With the organogenesis, of course, you get the development of the organ systems. But they don't always develop immediately in unison. So we'll see that blood vessels start as blood lakes. So they're little lakes of blood with developing cells surrounding them, and then eventually they become all continuous and then become the vessels, which are organs of the cardiovascular system. So there's that distinction between organs and organ systems. Organogenesis is occurring just a step or a stage before organ system development. Definitely at fetal, from 8 weeks on, everything is established. All the organ systems are in place, and there's a rapid growth and maturation of the organism with the organ systems occurring. There's still some final changes from the pluripotent to tetrapotent, dipotent, unipotent in that cell differentiation lineage that we talked about last time as well. Remember, there's the totipotent. This cell can become any kind of cell. There's pluripotent. This cell can be differentiated into many different kinds of cells. Multipotent, which isn't used very often, and then tetrapotent for 4, tripotent for 3 lines, bipotent for 2 lines, and then unipotent, it's going to differentiate into what it will become. So there's that whole series of events. So there's still a bit of that can occur in the fetal stage, but by far, most of the commitments to the unipotent stage have been made. In infancy, we see the newborn to the 2 years, and so our kids would always say, I'm not a baby anymore, I'm an infant. I don't know how they discovered the definition, but they sure did. So there's even some distinction there. Childhood, adolescence, adulthood, and then the later years we're all still very quite familiar with. What's the difference between adolescence and pubescence? Anybody take a psych course? What do you study in psychology? Adolescence or pubescence? So pubescence is the psychosocial aspect of the individual experiencing adolescence. Adolescence is the biological changes, the conglomeration of biological, physiological events that accompany it. So embryonic development is the first week of the first during embryonic development, we can look at the first week of the first trimester. So we know that basically we're talking about a 9-month gestation period in humans. Push the wrong button. Push the wrong button again. Pip on, pip off. Okay. We will now cover some of the major events, and I'll kind of toggle back and forth between the PowerPoint slide presentation and that website. So the diploid zygote divides. This is the state of cleavage. So we know that we'll study gametogenesis and see how the haploid gametes are produced. And then, of course, upon union of the gametes, we end up with a diploid zygote produced. So we will be changing the titles a little bit as we go along, but still it is zygote for the specific stage term all the way up to embryo. So the zygote at one point is going to be a ball of cells where it will look like a mulberry, and it is referred to as the morula. So we'll see there's a few different terms for stages of the development through the zygote phase. They'll become a hollow ball with the inner cell mass at one pole, and this is going to be called the blastocyst. So anytime you see blast, you know you're getting to the point where cells are closer to their differentiation, at least that initial step of, well, you're going to become the embryo and you're going to become the placenta. So it's at this stage that we begin to see some of that predetermination, and we have slightly more oriented differentiation lines or limited differentiation lines for the cells. I was just looking at those lights there, but if I turn that on, one's going to blast the screen. So you guys can't see it here, but the pip and just basically you can see my tie down to there. You can't see my face, but that's all right. Kind of a waste of a pip. The cyst, of course, refers to the hollow part of it, so there is an actual fluid-filled cyst within this structure. So terminology is going to be very important, as we had stated. Implantation within the endometrial lining will occur. Even before an individual might individual female might miss a menstrual period and begin to suspect pregnancy, implantation will be occurring. We will study the menstrual cycle so that we can understand those aspects of human development as well. But as we all know, in the basic model of the menstrual cycle, day 14 is ovulation, and the earliest pregnancy tests are within a week or so of when the menstrual flow would be expected. So that actually would be about 21 days from when the ovum is available for fertilization. So we're within the first week here, so an individual might even be unsuspecting that they are pregnant during this time, these series of events. So any of these cells are candidates for stem cell research and or cloning experiments, as we had stated, so it's important to understand this general term stem and how it applies to blast cells, stem cells in the bone marrow, and last time I even mentioned stem cells that a dentist had taken from a baby tooth of his own child and was able to cell culture and stimulate them to differentiate into different cell types. So certainly those are farther along in their determination. They're very limited, much more limited potentiation or potential for differentiating into different tissue cells, but still a valid source of that. So why dissociate an entire embryo to produce something that you can produce just by collecting baby teeth or maybe even tooth pulp from an adult? So we have to be creative in our thinking. The timetable, we can build on that some. Day one is the fertilization process, so the day of conception is the true day one of this new diploid organism. Day two is the two-cell stage, and you'll see that I'm very careful not to refer to the conceptus as a baby. A baby is the term for the newborn. Before that, it is either a fetus, an embryo, a collection of cells, or an organism. These are the terms that I restrict myself to in this classroom setting as a biologist. And it's very important. This is a setting in which we restrict our terminology so that we train ourselves in the discipline of biology. When I get home and we're talking about it, or talk to Jennifer in the hallway, probably going to get sick of me referring to you, but you can just tell me to stop privately sometime if it bothers you. You know, it's how's the baby doing? I'm not going to say, well, you know, how is the zygote doing or how is the fetus doing? you're going to ask her, how's your baby doing? And still kicking, you see her, you go, oh, my God. And you know that the baby is kicking, right? So we do make those distinctions. So we're practicing that level of discipline. Within the context of biology, we restrict our terminology. And again, it's because this is embedded within a very controversial setting within our society, the world society. So very quickly we begin to get cell division. It takes about a day for things to get organized. You know, we've got to have fusion of the pronuclei, and the chromosomes, the haploid chromosomes have to find each other and then call in all the proteins that signal it to uncoil and replicate, and then begin the process of making new cells. So DNA replication has to occur. So it takes about a day to get geared up, and then the 2-cell stage is there within about 30 hours, 4-cell in about 10 hours. So once it gets geared up, then it can start going very fast. If a cell falls off the cluster, it can develop into another embryo plus the placenta. In other words, this is one potential mechanism of generating identical twins. Day 3 is the morula. That's that solid ball of cells. Day 4 is the early blastocyst. So here we are. Day 4 is about day 18 to 20, let's say, or day 16 to 20, because ovulation can change. The only definite phase length is after ovulation if there is no pregnancy. The post-ovulatory phase is 14 days. That pre-ovulatory phase can vary from individual to individual and from cycle to cycle, which of course is just the point we're emphasizing, and we'll study that mechanism, the reason for that in detail when we study the menstrual cycle. But if we go with that model of day 14 ovulation, by day 18, the early blastocyst is produced, and it has that central cavity. They're now committed to becoming either placental cells or embryo cells, even though they really can't be completely recognized as such. They're just called an inner cell mass, and we expect and predict they're going to go in a certain way. Break into week 2, so and correct my math when I get off. I remember I couldn't add for beans on my feet the last semester I taught this. So jump in and help me. So this is 14 plus the 14. So now we're extending from say day 21, sticking with the 14-day ovulation, day 21 of the menstrual cycle to day 28. Day 28 would be, if someone is on a 28-day cycle, that should be the day of the last day of that menstrual cycle, and just a day before the first sign of menstrual flow, which is the overt sign that that's day 1 of a new menstrual cycle. So just on that date when someone might be expecting, well, any day soon, it should be starting my menstrual flow, that is the time point when the zygote as a blastocyst is already within the uterus, and it's beginning its process of implantation. It's also at this time, of course, that the cells that are now predetermined, so there's enough differentiation of the genes so that a hormone is being produced by these cells that make the outer containment of the blastocyst. They start producing human chorionic gonadotropin. And what does human chorionic gonadotropin do other than kill the rabbit being used for the pregnancy test? What does the human chorionic gonadotropin do? It promotes the corpus luteum, which is producing vast amounts of progesterone, which promotes gestation. And so we'll see, and we'll study the neuroendocrinology of this, we'll see how the developing zygote becomes independent of the maternal pituitary gland to sustain the pregnancy. And then eventually, by exploiting the maternal gonad, the ovary, the corpus luteum that's present, then eventually just becomes independent and controls the rest of your life, if you want to think of it that way. So the HCG targets the ovaries, as we said, and sustains progesterone primarily, but as well estrogen, and it will impact the pituitary to make the pregnancy independent of the maternal pituitary gland. The extraembryonic membranes, the amnion, the placenta, and the yolk sac are starting to develop, and the gastrulation process will begin. So gastrula makes us think of intestine. So gastrulation is going to lead us to believe that we might see some of the primordial gastrointestinal system appearing. And indeed, the yolk sac is a key part of that and is representative of that. It's at this time we get the germ layer development into a two-layer, and this terminology is wrong. It's a epiblast and hypoblast layer. We'll correct that as we go along. And then finally a trilaminar layer. This, by definition, the three-layer is embryonic. This, by our terminology, is inaccurate. The bilaminar embryo is the bilaminar zygote. It is not an embryo until it is three-layered. So that's the strict definition that we will use, and we make that point by presenting an inaccurate text on a slide. So day 6 to 7, as we continue on, I didn't toggle at all, did I? So this site lets us look at the post-ovulatory weeks. So week 1 is here, and it will open stage 1 website. So this is a very useful website. You can go, it offers a lot of information. It'll be a nice break from the text. It goes a little bit faster on my Roadrunner, so I might not be jumping to it all that frequently during lecture. And it offers really very interesting illustrations. So we see the appearance at 1 week, the second polar body is present. Why do we see the second polar body at this stage of zygote development? Because the second meiotic division is not triggered until just around conception. We see the two pronuclei are beginning to fuse. So the details are included here, and it gives you the actual dimensions of this. So it really provides a tremendous amount of information. I've enjoyed this site tremendously, so you should visit it whenever you can. So we are getting to the end of the second week, so I'm just going to go straight to week 3, and we see that we have the blastocyst is present here. This is the inner cell mass. It may be very close to forming the two-cell layer zygote. And you see the endometrial lining here. It is approximating it and getting ready for implantation. So I'll go back and forth to that every so often. So day 6 to 7, the implantation starts. We just saw that. We saw a polar arrangement of the cells. So the inner cell mass was actually located on the pole of the egg. Excuse me, not the egg. The zygote where implantation is going to occur. Why would that be preferential? What is the mechanism by which nutrients are reaching these highly metabolically active cells? It's all diffusion. So up until implantation, the source of the nutrients are the fluids within the fallopian tube lumina and the uterine cavity. So immediately upon contact with the uterine wall, the more highly concentrated secretions of the endometrium are present, and then with infiltration, ultimately there's a linkage to the maternal blood flow, the hemochorionic connection that we mentioned before. So I stated what we want to keep in mind is that these are very active cells. They're consuming, as far as the small number of cells is concerned, a large amount of energy to grow so rapidly. And of course, as those numbers of cells increase, there's still a very rapid metabolism and a tremendous energy demand on the mother. And of course, this is why we always hear you're eating for two, so you eat more when you're pregnant. So the day 8 is the two layered discs. So we said that in the previous image, we're very close to forming the 2-layer. 8 to 12, the conceptus gradually covered by the maternal tissue. So there's implantation with the gradual surrounding. The placental bed of vessels on the maternal side are going to expand locally. So there's a tremendous amount of communication between the cells. So the uterus knows exactly where implantation is occurring. So there's chemical signaling that is going on. And of course, we don't deal with that in any great degree, but we try to remind ourselves whenever we can exactly what's going on at that chemical level of structural organization. Day 13, some of the utero-placental circulation will begin. So let's go and take a look at week 4. Week 3 didn't really seem to go along with the timeline that we were just looking at. Here we see week 4, so this must be at the, it's stage 4. Yeah, so stage 4 is not week 4. Sorry about that. Stage 4 is the early part of that implantation. So now that I've read the legend, this is week 2 that we're in here, and week 3, this is the stage listed along here, so let's take a look at this. We can see that the outer cells are beginning to infiltrate into the endometrium, and the blood lakes are actually starting to form. So that utero-placental circulation is beginning to become established. So there's a lot of chemical signaling that's going on. There are a lot of hormones that are released to dissolve blood vessels on the maternal side, to make connections, tight connections. On the fetal side, the embryo side, we're very close to embryo, so I'll just use that term, on the zygote side of this, so that it doesn't allow the leakage of the blood in any type of hemorrhaging. So let's just go back to the slideshow and push on through this. So week 3, neurulation reaches the head region, so we're going to begin to see the development of the nervous system. We don't see the neurulation until the 3 layers form, and then some other major events. And we'll obviously be focusing on all the details of this, so we're just really completing the survey of this today. The embryo, this is not week 6, so this terminology is wrong, so we can correct as we go along. Looks like other chordates, fish, frogs, reptiles, birds, and other mammals. The HCG signal is stronger, so week 3, that's 21 days plus the 14, we're about 1 week past day 28 when an individual would have expected menstrual flow. So if unexpected, there would be some concern expressed certainly. If expected, there might be some jubilation. I don't know if any of you have seen the advertisement on TV where they show the parents about my age, I guess, so not so elder, looking and waiting for the results of a pregnancy test, and then it comes out positive, and they turn to the restroom where their teenage daughter is there. So that one plays off that statement that I was just making, individuals expecting or not expecting. So significant impacts. So understanding the biology of this is key as we become parents and responsible individuals, not only for being experts in biology, but in our own lives and in assisting others to understand what's happening. So the targets continue, tremendously high levels of progesterone, sustaining the endometrium, and then many other hormonal effects occurring to promote development of the mammary glands for the postnatal lactation. So in this class, it may be more allowed to comment a bit. As I said, Jennifer was in the summer research program, and she was studying endoplasmic reticulum and its response in neurons to decreased levels of ATP, and was kind of using the changes that we've all noticed over the summer in her body shape as the summer progressed. And we were talking about how her abdomen has increased, and that would be a good place to measure to get an indication of how the body changes during pregnancy. And then I mentioned that that would be compared to a measurement around her arm. And she says, well, why around my arm? I said, well, I didn't want to mention any other body parts that were growing, but she certainly is in a very healthy pregnancy, it seems, and has the glow of being a pregnant mom. So day 14, embryonic disc forms, as we had said. We see the primitive streak. So there's tremendous changes. So now what we realize is that we're not getting a simple division of cells to create multiple layers of cells. Now we're getting cells clustering together and undergoing mitotic division so rapidly that they're going to move as a solid column or solid ball of cells, leaving behind a column of cells that it divided and left behind. So a tremendous amount of change that's going to occur so that we see a streak that will form where this event is occurring. The day 15, the three-layered embryo disc is completed. So this is telling us that things are happening on a continual temporal basis, and we're trying to do freeze frame and say, well, at this day something is happening, at this day something is happening, at this week something is happening. When we think of the actual cytogenesis and all the gene changes and chemical signaling that occurs, it's an ongoing series of events, and the formation of the primitive streak doesn't just sit back and wait. Well, do we have the complete three-layered embryo formed yet before we can go on and do what we need to do? There's an anticipation that the three-layer formation is adequate enough so that a cluster of cells is triggered for some reason. It might be just temporal, or it might be chemical. It might be based upon the size and the changes in the supply of the nutrients, because we know that the uteroplacental circulation is becoming more established. Some signal tells this location of cells, it's your turn to undergo this next stage of differentiation and just dip in and begin dividing so rapidly that you produce a streak visible from the dorsal surface of this structure. So we're trying to piece it together into something that we can put together in our minds, and as I said, it's an exercise of audio and visual to produce some type of three-dimensional structure within our mind that will, I don't know what that was, that will be very useful for us in the future. So this notochord is what I was talking about. It's an axial scaffold later replaced by the spinal column. So very early on, this column of cells is going to be associated with the formation of the primitive streak, and it's going to contribute to the symmetry of the organism. It migrates toward the rostral pole where the head is going to form. It creates a split in the midline, so we've got right and left. It thereby also creates the other pole, the caudal pole, where the hind structures are going to develop. So the top layer of the ectoderm forms a central flat strip called the neural plate. Now we're getting a plate of cells dividing so rapidly, pushing laterally, and there's no place for it to go because there's a concentric constriction formed by two extra embryonic structures. And so if you can't get your rug to flatten out all the way in a room, it's going to crumple in a couple of spots. And where it crumples forms the neural plate. And then the nervous system precursor is identifiable. So the three germ layers are the ectoderm on the surface. It will reveal neurulation as the groove that we just described, the crumpling in the carpet that doesn't fit the room, will continue to bulge up until the tops of the two crumples come together and fuse and form a tube. and that'll be the spinal cord and the brain of the CNS, and it will leave behind the epidermis of the skin. Mesoderm is going to contribute a lot of stem cells forming the cardiovascular system, internal organs, all the connective tissue, including the bones and all of the musculature. So we will become experts through this semester at the products of each of these layers. The endoderm forming the gut, the lining of the gut actually, just about all of the GI tract and many of the associated organs and glands. So now we're at day 20 with the middle layer of the disc, the mesoderm, making that final third layer, and it'll partition into segments called the somites. So there's going to be these extremely visible changes that are occurring. The neural plate rolls into the tube and then will disappear under the surface of the ectoderm and go on to form the spinal cord and the brain. Days 21 to 22. So this is 3 weeks from ovulation. The conception will occur typically within 24 hours. The ovum is viable in the female reproductive tract for up to 24 hours. And the sperm, by the way, for 48 hours. So there is still some potential for overlap there. So 3 weeks later, the heart is already beating. It is a simple tube, of course, but it is already lined by cardiac myocytes, and they are displaying autorhythmicity as do the adult cardiac myocytes. And then other vessels and blood cells will be forming all over the place, as we had said. So we'll stop here with week 2, and we'll complete this on Friday and go straight through. I'm going to go back to the what's this called? The Elmo, and project up here the name of the 1604 location, distance learning proxy, forgotten the actual term. Anyway, a graduate student in the Master's of Biology program, Marco Riojas. Remember, I'm a Yankee. You know every time I say your name. His email is shown here. He's an alumnus of MIT, you can tell. And we'll have another individual at downtown as soon as we can. I'm sorry, I didn't get that for the students downtown. Nayeli Ayala is designated. She'll start coming downtown to be there to help out. I'll be at Brooks. Marco will be here. Nayeli will be downtown. And this will be our model that we're developing with the use of the student fees, which I'm sure that you've noticed in the College of Sciences, to help interface with you and meet your needs in a variety of mechanisms. This is just one. Marco, you can touch that and you'll be on the camera, so they'll be able to see you downtown. Wait until it should zoom in. It didn't zoom in. So he's here at 1604, and then Nayeli will be very soon with you downtown. I appreciate your time and attention. Apologize for being late. We'll pick up where we left off on Friday.